Hello everyone. Today I'm gonna talk about my work on examining the interaction between free living bacteria and iron in the global ocean. So in the ocean, bacteria play an important role in ocean biogeochemistry due to the fact that they can transform the organic matter that fixed by phytoplankton in the surface ocean into a refractory form and keeping carbon away from the atmosphere for thousands of years, or they can remineralize some part of the organic matter that fixed by phytoplankton in the surface ocean into inorganic forms, thus maintaining the ocean primary production in the surface ocean, or the bacteria they can transfer organic matter up the food chain because they can be grazed upon by zooplankton. So it's very important to understand the factors that limiting the growth of bacteria in the ocean and how bacteria impact the ocean biochemical cycles. In this study, I'm gonna focus on the free living carbon, carbon oxidizing bacteria, but there can be many types of bacteria in the ocean. The particle attached bacteria, as well as the nitrogen and sulfur oxidizing bacteria as well. In the ocean, free living carbon oxidizing bacteria, they require dissolved organic carbon and iron for their growth. So there's a potential competition between bacteria and phytoplankton for iron. And as shown by Totel et al. In 1996, by some field studies in the North Pacific Ocean, the iron content and iron requirement by free living bacteria usually are higher than the iron content and requirement by phytoplankton. So again, there is potential competition between free living bacteria and phytoplankton for iron, and that's why free living bacteria can affect the ocean iron distribution through their growth and mortality. And because bacteria do not require light, they can live everywhere in the ocean. So that's why free living bacteria can impact the ocean iron distribution in the surface as well as in the subsurface, as shown in the figure here. And second of all, some studies also show that free living bacteria can also produce ligand, which is the organic matter that can form complex with iron to protect iron from scavenging. So overall, my research question in this study is how free living bacteria affect the ocean ion cycling through their activities in the ocean. And to do that, to explore the free living bacteria ion interaction in the ocean, I'm going to use an ocean biogeochemistry model, basically the Nemo-Piscis model has that results from nanophytoplankton diatoms and has two zooplankton components, the micro zooplankton, the mesozooplankton, and also solves for the cycling of nitrate, iron, silicate, and ammonium in the ocean. And for this study, I resolved the bacteria, free living bacteria component in the model. And I also resolved the iron content of free living bacteria in the model. In the model, free living bacteria require iron and labor dissolved organic carbon for their growth. And, and when they die, they also release dissolved organic carbon back into the ocean, and then they can be grazed upon by microzooplankton. So what I what I first need to do is I evaluate the model results by looking at the distribution of the surface model bacteria biomass on the shown on the left here, we see that in the surface, free living bacteria is high in the subtropical Pacific Ocean and also in the North Pacific and North Atlantic and Southern Ocean. And if we compare the vertical profile of free living bacterial biomass in our model, average over the global ocean and average over the tropical ocean, and compare that with available observations from Maradat data shown here on the middle figure here. The full line is the results from the model, and the dotted line is the results from observations. We see that the model free living bacterial biomass is in the range indicated by observations. And finally, on the right hand side here is the ratio of free living bacteria production to primary production in the surface ocean. And this model results, we see that bacteria production is high in the subtropical and tropical oceans. And this results is consistent with what we know from observations. So to analyze the factors that limiting the free living bacteria growth in the ocean, I analyzed the limitation status for the growth of free living bacteria first in June, July, and August by looking at the map of the surface limitation status for bacteria by DOC, dissolved organic carbon, and by iron. And basically, between DOC and iron, the nutrient that has lower value for the surface limitation status for bacteria is the nutrient that limits the growth of free living bacteria in the ocean. And we see that over most of the ocean in June, July, and August, DOC is the ultimate limiting factor for the growth of free living bacteria over most of the ocean, except for some regions in the North Atlantic, North Pacific, and tropical Pacific. We do the same thing for December, January, and February. And again, we see that over most of the ocean, DOC is the ultimate limiting factor for the growth of free living bacteria in the ocean, except for some regions in the Southern Ocean, Eastern Pacific, North Pacific, and tropical Pacific. So we can conclude that over most of the ocean and over most of the year, DOC is a fundamental ultimate limiting factor for the growth of free living bacteria in the ocean, with iron can be the secondary co-limiting factor for the growth of free living bacteria in low iron regions, such as the Southern Ocean, North Pacific, and tropical Pacific. And if you look at, you want to look at the impact of free living bacteria in the ocean ion cycle. First, looking at the global scale here, averaging over the global scale, we see that first in the surface ocean, the amount of iron consumed by bacteria in the surface ocean is around 50% of the amount of iron is consumed by phytoplankton. And second, the amount of iron that zooplankton get from bacteria is also about 50% the amount of iron that zooplankton get from phytoplankton. And also in the surface ocean, the iron fluxes induced by bacteria, the free living bacteria growth and mortality are really significant. But in the subsurface ocean, in the deep ocean, surface ocean, under the thermocline, lower than the thermocline level, the iron fluxes induced by free living bacteria activities, specifically the iron consumed by free living bacteria and the iron released from the mortality free living bacteria, their values are in the same order of magnitude. So these two values are really close to each other and they kind of cancel each other out. And the net iron flux induced by free living bacteria activity in the thermocline are really small compared to other fluxes of iron in the subsurface ocean, like the remineralization flux and the scavenging and coagulation flux. And if you look at the distribution of the iron uptake by free living bacteria and iron released by free living bacteria mortality in the ocean, we see that the iron flux is induced by bacteria activities in the surface ocean are really significant. And the second thing is the iron consumed by free living bacteria are higher than the iron released by the mortality of free living bacteria in the surface ocean. So in the surface ocean, free living bacteria as act as a sink for the iron in the surface ocean. And if we look at the contribution of free living bacteria to the total biological uptake of iron in the aquatic zone, we see that free living bacteria can contribute to around 30 to 40% of the total biological uptake of iron in the aquatic zone in the Southern Ocean, subtropical Pacific, and North Pacific. And that's why, again, free living bacteria are significant competitor with phytoplankton for iron in the surface ocean. But again, if we look at the distribution of the net flux induced by bacterial activities in the subsurface ocean and compare that with the main source and sink of iron flux in the subsurface ocean, which is the scavenging and colloidal iron flux in the subsurface ocean, we see that the impact of free living bacteria on the subsurface ocean is very small, due to the compensation between the iron consumed by free living bacteria and iron released by the mortality of free living bacteria in the subsurface ocean. So to conclude, iron emerges as approximately factor that limiting the growth of free living bacteria in low iron regions, with labile dissolved organic carbon play a fundamental role and is the ultimate limiting resource for the growth of free living bacteria over most of the ocean and throughout the year. Second, free living bacteria consume a significant amount of iron in the surface ocean and can compare and can compete with phytoplankton for iron in the surface ocean, but their impact on the subsurface iron cycling is small. However, in our study, we only consider the free living carbon oxidizing bacteria. So there are many types of bacteria in the ocean, and like as I said before, the nitrogen oxidizing bacteria, the sulfur oxidizing bacteria, and particle attached bacteria, they all can play an important role in the ocean ion cycling in particular and ocean biochemistry in general. And that's why a more complete understanding of the role bacteria has to be considered in the future. And we need a holistic consideration of the role of different bacterial groups and as well as their specific ion requirements, demands, and their ion, their capability to store ion in the cell to have a better understanding of the role of bacteria in the ocean ion cycling. And second of all, as I mentioned before, we lack observations for the distribution of bacteria biomass in the ocean and observations and measurements about bacteria iron requirements and demand and capability to store iron in the cell to really constrain the model results and to know how to exactly represent bacteria and their interaction with iron in the ocean biochemistry models. So that's going to be the main focus of future study, and our study definitely not the last study, and just one step forward in representing bacteria and their interaction with iron in the ocean biochemistry model. Thank you for your listening and for your attention. I'm looking forward to talk with you more during the conference. Thank you.